Assalamu alaikum students. This is Tabassum Rahman Sanfi, lecturer of Biotechnology program, BRAC University. And I will teach you for the course Introduction of Biotechnology and Genetic Engineering, which is BTE 101. Welcome to my course. Well, as this is our first class, so this might be a practical meetup for us at the university. But due to the pandemic of COVID-19, we are going to start the semester summer 2020 at a different way, and I hope all of you are in good health. Before starting the lecture topics or starting the bookish procedures, I will give you a brief idea about the topics or what we are going to learn from this course. And I can assure you that this is one of the most interesting topic or one of the most interesting chapter in biological science. From the name you might have an idea what is the basic theme of this course: genetic engineering and biotechnology. That means we'll learn about the genetic constituents, how to manipulate them, how to apply engineering knowledge in case of changing our genetic code and creating some kind of biotechnological products from them. In the first lecture, I will give you some brief idea about some common terminologies, and also will discuss with you about the most important discoveries in the era of biotechnology and genetic engineering. With a positive mindset, let's got into the lecture. Well, the first term we will learn here for this course is the term that is mentioned in the name of the course or main name of the topic title, which is biotechnology. Well, what is biotechnology? The name itself is a combination of two different words. One is biology, another is technology, which clearly refers the application of biological science in order to come up with some technological advancement. That means you need to know how and where you can manipulate all the biological sources, organisms or substances retrieved from them in order to have some technological advancement. Here you can see a very clear definition, which is biotechnology involves scientific study. Of course, as we are living in the era of science, we have to proceed with scientific study in every aspect. So, biotechnology involves scientific study and manipulation of living organisms or substances from them. Just look at the point. This involves the scientific study of either manipulation of living organism, that means modification or changes, how to occur changes in either living organism or substances from them. This might be an enzyme or other components or other living things to solve problems or make useful products. Now, this is a very clear definition. Please keep in mind this type of definitions are very much important for both your MCQ question answer, short question, broad question, or most importantly to clear your basic concepts in order to have a firm idea about biological science. Now, in the second slide, we'll know about a most common term. We are hearing, we are learning, or even we are seeing this word frequently in our day-to-day life. We all are saying that science is doing this, science is doing that. So the help of science, we are having this. This is the negative or positive effect of science. Every time we are uttering this word science, but I think hardly few people will know the basic definition or the main concept of science. What is this science that is inventing or that is nourishing the whole world, that is nourishing everything or that have impact in our everyday lives? It is nothing but a systematic knowledge. Science itself is a knowledge, but in a systematic way. In order to predict something, in order to imply one of your idea, you have to go through some systematic approach or systematic experiments. Further, your output will be analyzed in order to justify whether your idea or your prediction about a certain event or a certain phenomena is actual or not. So in an easy sentence, it has been said that Scientific observations, of course it is observations, are systematically conducted on purpose and the results are recorded. That means you have to observe something and you have to have some specific purpose, then that for this you are willing to do this. You want to invent something, you want to have a certain goal or a certain aim, and keeping in mind about that certain aim you have to proceed forward. This is called science or scientific studies. Science gives us the power to predict events through science or through applying the systematic knowledge in order to conduct a certain work or certain approach. You can have an idea or you can have the caliber to predict something, that whether it is right or wrong, or how much sustainable that particular event for us or not. So this is science. So we now know two different definitions about two different terminologies. One is biotechnology, another is science. Again, the repetition of the definition, which is biotechnology, involves use of living organisms or substances from those organisms to make or modify a product. That means you can either create new organism or modify a product of them in order to improve your plants or animals or to develop microorganisms for specific uses. Step by step, we will know about all the applications in our upcoming lectures. We are uttering the word biotechnology. It is a combination of two words, but we must know about the scientist or the person who first coined the term biotechnology. It was in the time of 1919, and a scientist named Karl Erecki, an Hungarian engineer. Look, he's not a biological scientist, he's an engineer. He's not even a scientist, he's just an engineer, but he first coined the term biotechnology. Until now, from 1919 to 2020, it's been almost 101 years. We are still using the word biotechnology at our everyday purpose. It is one form or another has flourished since prehistoric times. That means from prehistoric times, biotechnology has some significant impacts in our daily life. Traditional biotechnology, that means the biotechnology before it is modernized. In the traditional period, traditional biotechnology has been used for thousands of years to produce improved food and healthcare products. That means from ancient period we know the application of biotechnology in order to have improvised version of food products or other healthcare products. Now what does modern biotechnology do? The basic difference between traditional biotechnology and modern biotechnology is that the modernization of biotechnology has given us to ensure the safest and rapid way of these pre-existing procedures. That means modern biotechnology also try to have improved food or other healthcare products or other product compounds, but in a safer way. This slight difference has changed the whole system from traditional to modern. That means now we know how to improve a crop but in a safer way. But previously it was known that only through biotechnology we can have a specific procedure to have a novel compound or modified compound. But now we know what is a safe approach to have that particular type of modified crop, food product or other things. So this is the basic differences. Classical examples of using biotechnology. I think all of you are familiar with these examples, like making bread in brewery industry or fermenting grapes to produce wine. Although this is not very common in our country, but in European country and others, fermentation of grapes and other fruits or other vegetables to produce wine is very common. For us, using yeast to make bread or even dough, pizza dough, not only loaf but also different other types of bread, nowadays we produce in our house using yeast, either dead or live. Another is domesticated plants and animals are the result of selective breeding. What does it mean? It means we know how we can domesticate plants or animals to have a better breed. For example, a tomato or a potato. which is growing in land, but the yield rate or the size of the potato is not up to the mark. So what we can do, we can have some selective breeding. That means we can take two different types of potato plants and can further make them crossing to have a better breed. This is a very common approach in previous type of biotechnology. And these are the classical examples. Now let's see. Throughout the period of 1919 or even from before that to at this period of 2020, all this journey have been divided into three different sectors. And through this, we can have the idea about the development of biotechnology. At ancient period, people used to produce bread from yeast, wine from rotten grapes or other vegetables, but they have merely any idea that biotechnology was involved there, and that was ancient biotechnology. Biotechnology was there, but it was not visible or it was not known to the personnel who is conducting or who is responsible to conduct such reactions. Now, see the examples. Ancient biotechnology, early history relates to improved food by domestication of plants and animals. That means this is an example of ancient biotechnology. Please point out these examples. These are very important for all types of questions. Example of classical biotechnology, built on ancient biotechnology. That means they are built on the theme of ancient biotechnology, but with an improved version. That means fermentation, promoted food production, and medicine. These two are example of classical. Now, what is modern biotechnology? Manipulates genetic information. That means now we know the specific point where we can make the change in order to have a desired product or a desired animal or plant, whatever you say. That means modern biotechnology relates the manipulation of genetic information in organisms by genetic engineering. So we can say that genetic engineering is the key of modern biotechnology. Please keep in mind, it is genetic engineering. That means engineering of gene level or changing the construction of genetic level in order to have something or having a manipulated product. And this is the key of modern biotechnology. Now the upcoming slides of the lecture you can have or you can see in the next portion of the video recording. Thank you.